Is that right? This is onion root tip. Everything else is interface. Most everything else is interface. Metaphase, most everything else is interface. Mitosis, of course. You have metaphase. This is cell plate. You guys see that between? This is another cell plate in here. So this is telophase. You cannot write down one or two. Nobody's sitting down. Do you want me? Can I sit down then? If nobody's sitting down, okay. So that's the one. Is it everything else is okay? Same thing. Just nice, beautiful cell plate, metaphase, telophase. Okay. In this magnification, I don't see any stages, right? So, but I might ask you what process occurs here. Mitosis, of course. So this is telophase because of the cleavage furrow. You guys see that? Cleavage furrow, chromosomes. In the last, and this is another nice telophase. But last few slides, I might ask you, what is the name of structure? Chromosomes. You should know that. These are all chromosomes. You're not looking at anything else but chromosomes here. And nucleus, of course, prophase. So telophase, anaphase, of course, another nice, beautiful anaphase, metaphase, prophase, everything else is interphase. Okay, lilianther, I cannot see any stages. I might ask you what is the part of the plant. I have been asking you on these quizzes. Anaphase 1. All of these are anaphase 1. Nice, beautiful anaphase 1. You do not write down 1, I will not give you credit. You should know, you see how the pollens, future pollens, they are apart from each other. So you should know you are in meiosis. Yes, all of the cells are compact together. Look like these, but no, they're all packed together, so it is mitosis. Of course, this is the meiosis. Prophase 1, metaphase 1, prophase 1, metaphase 2, telophase 1, telophase 2. There are 4 of them. They all will separate and become a pollen and goes inside of your nose, and of course you take antihistamine. I do not recommend that. I say just salt water. Okay, ovary, right? That was your question. What organ? Ovary. This is corpus luteum. This is Graafian follicle. And inside that is egg. What is the name of the cell? Egg. What is the name of the cell? Egg. What is the name of structure? Graafian follicle. What is the name of structure? That is corpus luteum. When the egg leaves Graafian follicle, the Graafian follicle becomes corpus luteum. It releases progesterone. Okay, testes. What organ? Testes. What process occurs here? You don't see it, but meiosis. What process occurs here? Meiosis. What process occurs here? Meiosis. You don't see the stages, but it does occur here. So, what is the name of structure? Seminiferous tubule. What organ? Testes. What process occurs here? Meiosis. What is the name of the cell? Sperm. These are all sperm. The tail of the sperm, the head of the sperm. You see that? Whole bunch of them. And then of course these, I didn't ask you to know the name of these when you take zoology. Sperm, head, tail, skin. What process occurs here? Mitosis. We did not have. Hydra, we do have it in the lab. The model, I did not bring you the slide, so do not worry about the slide. But this is budding or mitosis. Budding is a type of mitosis. So this one becomes just like mother hydra. It will separate from mother hydra and sits on the bottom of the lake. Stages of mitosis, interphase. They are not stages of mitosis. These two are not stages of mitosis. Of course, this is the S phase of interphase. Prophase, metaphase, anaphase, telophase, fertilized egg, which is called zygote, 2 cells, 4 cells, 8 cells, morula, blastula, gastrula. We do have a different model. Same thing. In the gastrula, you have invagination. These, it's just a ball of cells inside of it is empty. Blastula. Morula is a ball of cells, inside of it is full of cells. Gastrula, there is an invagination right here. They call it invagination. Unfertilized egg, morula, gastrula, invagination right here. When you take zoology, we will go over more details. Blastocyst or blastula, same thing. Gel electrophoresis, what is the name of, this is centrifuge, mini centrifuge, is that right? We talked about it. This is the PCR machine. So I might put that in there and say, what is the name of the equipment? PCR. Again, none of these you have to worry about. The gel electrophoresis machine, right? What is the name of the equipment? Gel electrophoresis. Okay, these are, we talked about them. I don't think this is the vortex machine, right? Again, mini centrifuge, again, none of these are, well, we did talk about these guys, right? We did talk about this today. Capital A, capital A, capital A, small a, small a, small a. And then these two guys are, there should be another one, but anyhow, there's not one. None of these, well, the criminal, you guys know, we talked about it before. Blood typing, of course. So what else? Corn is coming up. Corn. What is the phenotypic ratio? Is 9:3 to 3:1. What is the parents of this? Big A, small a. Big R, small r is one parent. The other parent is big A, small a, big R, small r. Right? Same thing here. What is the kernel? I'm at point at the kernel. What is the genotype of that kernel? Of course, it's yellow, so it's small a, big R, blank. Do you guys follow what I'm talking about? Big R, blank. The second one, whatever it is, doesn't matter, big R. But this one is yellow, right? So it's small a. You cannot write down small a blank. Well, then the blank, I say, I will argue with you, it's big A, which is not right. If it is yellow, it is small a. All of these, all of the yellow ones, small a. The dark one, they are big A, right? Am I? Color, yeah, color. So big A, blank. Anyhow, let's go. 9:3, 3:1. This is 3:1. Phenotypic ratio is 3:1. I don't have any sugar here. Everything is starchy, so it's 3:1. The parents, big A, everything is starchy, so I don't have to say anything about R's. Big A, small a, and one parent, and big A, small a, another parent. So the parents in here is phenotypic ratio 1:1 is an example of a test cross. So one parent is big A, small, big R, small r, and the other parent is small r, and everything is small a, right? You guys know what I'm talking about. Enzyme lab, we did not do cellular respiration, was different, and that's it. I went